Hello everyone. How do pharmaceutical laboratories determine the concentration or assay of a drug accurately? One of the most fundamental technique is titration. In this video, we will understand the principle, types, procedure, endpoint, calculation and pharmaceutical applications of titration. What is titration? Titration is a qualitative and quantitative analytical technique in which a solution of known concentration called the titrant is added to a sample solution until the reaction reaches its endpoint. From the volume of titrant consumed, the amount or concentration of the analyte can be calculated. Basic principle. The basic principle is based on a chemical reaction between the analyte and titrant. At the equivalence point, the reacting substances are present in their stoichiometric proportion. In practical analysis, we detect this point using an indicator or an instrumental technique. Important terms. There are several important terms to remember. The analyte is the substance being analyzed. The titrant is the solution of known concentration. The burette delivers the titrant accurately. The endpoint is the experimentally observed point, indicating completion of the reaction, while the equivalence point is the theoretical stoichiometric point. Apparatus. The commonly used apparatus includes a burette, pipette, conical flask, volumetric flask, measuring equipment, and an appropriate indicator. In pharmaceutical laboratories, calibrated glassware and proper analytical practices are essential for obtaining reliable results. Preparing the sample. First, the pharmaceutical sample is accurately weighed or measured. It is dissolved using a suitable solvent and when required transferred quantitatively into a volumetric flask and diluted to the required volume. Filling the burette. Before titration, the burette should be properly rinsed and then conditioned with the titrant. Fill the burette above the zero mark, remove any air bubble from the tip and record the initial burette reading carefully. Pipetting sample. Using a calibrated pipette, transfer an accurately measured liquid of sample into a clean conical flask. Add the required solvent, reagent, or buffer according to the analytical procedure. Adding indicator. Now add the specified amount of a suitable indicator. If the method uses a visual endpoint, the choice of indicator depends on the chemistry of the titration and the expected endpoint. Performing titration. Start adding the titrant while continuously filling the flask. As the endpoint approach, add the titrant slowly, often dropwise. Observe the solution carefully and stop when the specified endpoint change is reached. Endpoint. The endpoint is the point at which the indicator produces the required observable change. For example, in some acid-base titration, a specific color change indicates that the endpoint has been reached. Reading burette. After reaching the endpoint, record the final burette reading. The volume of titrant consumed is calculated by subtracting the initial reading from the final reading. The burette should be read carefully at eye level to minimize parallax error. Calculation. The analytical calculation depends on the reaction, stoichiometry, and the concentration expression used in the method. A commonly used relationship is C1V1 divided by the appropriate stoichiometric factor equal to C2V2 divided by its corresponding factor. In pharmaceutical analysis, the final result may be reported as a percentage, concentration, amount of active ingredient, or another specified results. Type of titration. The major type of titration include acid-base titration, redox titration, precipitation titration, and complexometric titration. Acid-base titration involves proper transfer reaction. Redox titration involves oxidation and reduction. Precipitation titration forms an insoluble precipitate, while complexometric titration commonly involves complex formation between a metal ion and a legend such as EDT. Pharmaceutical applications. Titration has many applications in pharmaceutical analysis. It can be used for assay of pharmaceutical substance, determination of acidity or alkalinity, analysis of raw materials, standardization of solution, determination of certain metal ions, and quality control testing of pharmaceutical products. Accuracy precautions. For accurate result, use properly calibrated glassware. Ensure the burette tip is free of air bubbles. Read the meniscus correctly. Avoid overshooting the endpoint. Use a suitable indicator. Perform replicate titration when required. And follow the validated or compendial analytical procedure. Common errors. Common errors include incorrect burette reading, air bubbles in the burette, improper rinsing, contamination of solution, incorrect indicator selection, adding titrant too quickly near the endpoint, and incomplete dissolution of the sample. Final recap. So remember the basic sequence. Prepare the sample. Prepare and fill the burette. Transfer the aliquot. Add the required reagent or indicator. Titrate carefully to the endpoint. Record the readings. Calculate the titrant volume. And finally calculate the results. Titration may look simple, but accurate technique, correct stoichiometry, and proper analytical procedure are essential for reliable results. If you found this explanation useful, like the video and subscribe for more pharmaceutical analysis and quality control topics.